Hey guys, welcome back to our second video for the muscle unit. Um, Last time we had a pretty easy introduction into muscle types and functions, and today we're just going to dive headfirst into what is arguably the most difficult part of the chapter. And I'm not going to sugarcoat it for you. It's muscle physiology, and it is tough. But we can do it. It is going to be kind of a long one, so buckle up. We're going to cover a little bit of ground today. So here's the deal. We're looking at skeletal muscles here. This, at this point in your life, you should know is called a fascicle. It is bound by what's called the paramecium. They've pulled out of this one myofibril, which by now you know myo means muscle, fibril means fiber. So they've brought out one muscle fiber, which is the same thing as saying one muscle cell. Now, here we have multinucleate cells. You can see a nucleus here, a nucleus here, and a nucleus here. The nuclei are located just beneath what is called the sarcolemma. They give the plasma membrane a special name in muscle cells. They call it the sarcolemma within the paramecium. And so that's where we're at from a geography point of view here. We have striated muscle, a dark band here, a light band here. So the sarcolemma, like we already mentioned, is a specialized plasma membrane, and it also has within this the sarcoplasmic reticulum, which might sound a lot like endoplasmic reticulum, which you've heard of before in general biology, and that's a specialized smooth ER, and we're going to show you what that's used for here in just a minute. So the myofibril are these bundles of teeny tiny myofilaments. So within a muscle cell are little tiny muscle filaments or muscle proteins, and the myofibrils are aligned to give these distinct bands. Now, there might be a point in your life where you learn all the names of every band, but that point is not today. I want you to know that there's a light band and a dark band, and where the muscle fibers are located is what gives us the appearance of a light band as opposed to a dark band. And that's about all you need to know for right now. Alright, so our sarcomere, this is an important term, is the contractile unit of a muscle fiber. We have thin filaments, which are called actin. They're made of the protein, creatively named actin. And then we have thick filaments, made of myosin, made of the protein, creatively named myosin. Myosin's thick, actin is thin. These two fellows work together to make our muscles contract. And today we're going to learn how. So if you take a closer look at the organization of the sarcomere, we've zoomed way in now. You have your thick filament, which is right here. See this big red guy? That's called your myosin. Outside of it, you have these teeny tiny thin little braids. That's called your actin. Now, you can tell that light color and that dark color coming from where that really thick dark myosin is. And these guys have ATPase enzymes, which means they're breaking down ATP, and their job consumes a lot of ATP. And we're going to look at how that's used here in just a minute when we look at these videos. So that's the organization of your sarcomere. Thin filaments are actin. They're made of actin. Got it. Got it. So myosin, closer look. Myosin, actin. These little guys right here that look like a couple of balloons trying to float away are myosin cross bridges or myosin heads. Heads, cross bridges, whatever you want to call it. The actin and the myosin overlap a little bit. And what you're going to see in just a minute is these myosin heads are very important because they're going to grab that actin and they're going to pull it. And each one only pulls a teeny tiny distance, but there are many cross bridges, and working together they can do a lot. So all we're looking at doing is taking one protein and moving one protein a little tiny ways. That's it. That's all we're looking at right now. So there's two things that our skeletal muscles can do. A couple of their properties, there's irritability and contractility. They sound a lot alike, but they really are different. If you're irritable, you receive and respond to a stimulus. Think about a little brother, little sister, maybe a parent. If you say something to them and they don't have much of a response, if they're in a mood that you might describe as irritable, You say something to them, and they have quite the response, because they're responding to the stimulus, which is you bothering them. There's also contractility. Contractility is the ability to shorten when an adequate stimulus is received. Shorten. That's what contract means, right? So, irritable, you get a stimulus. Contractile, you shorten. Muscles receive a stimulus, respond to a stimulus, and shorten. And that is how a muscle works. Well, that's not how. That's describing how. We're going to see how muscles work here in just a minute. So how do you get a skeletal muscle to contract? The first thing you need to do is you need to tell it to contract. And in a human body, you get those through nerves. Here's a nerve, a neuron running out to muscle fibers, and another neuron running out to a different set of muscle fibers. Skeletal muscle has to be stimulated by a nerve in order to contract. And a motor unit is how many muscle cells are stimulated by one neuron. So one neuron can stimulate a couple muscle cells. This purple motor unit 2 right here, he ends at 1, 2, 3 muscle cells. Okay? Motor unit 1 is red. He ends at 1, 2 muscle cells. Not a big deal. So motor unit 2 will make these 3 muscle cells contract. Motor unit 1 will make these 2 muscle cells contract. And if you're lifting something that's, say, very, very, very heavy, you might very well fire both of these and make every single fiber contract. It would make you fatigue quickly, but you'd be able to generate a lot of force. So your brain sends a signal which travels down the spinal cord. It exits the spinal cord through nerves. The nerves go to the muscle. The nerves contact the muscle. That's not actually true. They get close to contacting the muscle. They send the neurotransmitter, the muscle contracts, and you've moved. That's how it works. So I said they don't really touch the muscle, and here's why. This is called an NMJ, a neuromuscular junction. The NMJ is also called a synapse. They're really the same thing. Well, maybe I shouldn't say that. All NMJs are synapses. Not all synapses are NMJs, something along those lines. This is a synapse right here. This is a neuromuscular junction. This guy up here is the nerve. This guy down here is the muscle. And what's going to happen is that nerve ends in this little tiny guy called a synaptic knob or a synaptic button or a synapse, something along those lines. And it doesn't quite touch the muscle. It looks like it does. Don't get me wrong, the muscle runs right up here, and it runs right up here. But the muscle actually has an indentation. And that synaptic button of the nerve sits right there, and he doesn't touch the muscle. And so this is a site where these two work together. And what's going to happen is when this guy decides to send a signal, he's going to diffuse out a neurotransmitter. And that neurotransmitter is up here in a vesicle. The vesicle fuses. You might have heard the word exocytosis earlier in your life and thought, what in the world, when is that going to matter? Here's your answer. These little vesicles merge with the plasma membrane in what's called exocytosis. They secrete a neurotransmitter. The neurotransmitter touches the muscle cells, and we've communicated to that muscle cell, hey, you guys down here, it's time for you to contract, and that's exactly what they do. So that's how we send that message. The synaptic cleft is what we call the gap. Synaptic cleft between the nerve and the muscle cells. So this little gap, let's see if I can do this with my mouse, right there is a space. And that space runs all the way down here. That's called the synaptic cleft. The nerve and the muscle do not actually touch, which we already talked about, and the area between the nerve and the muscle is, not surprisingly to you, filled with fluid. No duh. You knew that, I'm sure. So our neurotransmitter is a chemical that's released by that nerve when you get the nerve impulse, which is you thinking, move my leg or move my arm or whatever. And the neurotransmitter for skeletal muscle is one I want you to know. It's called acetylcholine. You don't have to memorize a lot of neurotransmitters in high school anatomy, but you do need to memorize acetylcholine. Acetylcholine is a neurotransmitter that the nerve releases and the skeletal muscle picks up. And that neurotransmitter attaches to receptors on the sarcolemma, a specialized plasma membrane that you learned about in our last lecture. The sarcolemma, when it gets that neurotransmitter, becomes very, very permeable to an ion called sodium. So sodium goes rushing into the cell, into the muscle cell, and it actually generates an action potential, which is kind of amazing because you'll learn this in nerves, that nerves run on an action potential, and currently you're learning that muscles run on an action potential. You can kind of think of an action potential as like transmitting an electric charge. It's not exactly like that, but it's close. And once you start, muscle contraction can't be stopped. Once that nerve says contract muscles and that sodium's in, it's in, and those muscles are going to contract, and you can't stop it. So, when you activate the nerve, it causes these myosin heads to attach to the thin filaments. These little balloon guys I told you about are going to reach up and grab actin. The myosin heads are then going to attach to the next site on the thin filament. What they're going to do is they're going to pull this thin filament this way, and they're going to pull this thin filament this way, just by grabbing on with these myosin heads. I'm going to show you that in a video here in just a second. When they're done, you'll see that the actin has been pulled in and is actually overlapping a little bit. And we've taken these little thin bands, the Z band, the I band, and we've actually shrunk them down to where they're almost non-existent. Here's the dark myosin proteins of the next sarcomere, the next functional unit. And then here, these guys together in that Z band and I band are almost completely gone. So all we're doing is dragging proteins over top of other proteins, which is a very small thing, but when you combine millions and billions of them, it becomes a very large thing. So this continued action causes a sliding of the myosin along the actin, and then the muscle is actually shortened. We have shortened because of these proteins coming in close together. We've made this muscle unit a little bit shorter, which makes the whole muscle just a little bit shorter. And that's what happens when muscles contract. So this is best explained by what's called the sliding filament theory, which is basically the idea that as calcium rushes into the cell, these myosin cross bridges are able to grab on to the actin. And there's a couple proteins that'll be stopping him this whole time, and we'll talk about those in the video. But he's going to come in and grab onto the actin, and he's going to pull him this way. Here you have him pulling actin this way, pulling actin this way, and that's exactly what happens. And then the muscle is quite a bit shorter now. So muscle fiber contraction is all or none. Think about it like a light switch that doesn't have a dimmer. Muscle's either contracted or it's relaxed. And you might think, well, geez, Mr. Stecker, that kind of contradicts what you just told me, that muscles were like maintaining my posture all the time. How does that work? Well, we also mentioned that nerves can go to a motor unit that might only be a couple muscle fibers. So just because muscle contraction is all or none doesn't mean that when you walk, you necessarily have to contract every single muscle fiber in every muscle of your quadriceps. You can just contract enough to get the job done. But in those muscle cells, that contraction is all or none. So hopefully that makes sense. Within a skeletal muscle, not all the fibers are going to be stimulated during the same interval, which is kind of what I just explained to you. Different combinations of these muscle fiber contractions can give differing responses. For example, walking as opposed to leg pressing in the weight room trying to max out. Those are two very, very different jobs, and they have different responses because of how many muscle fibers and which muscle fibers you're activating. We have what are called graded responses. Those are different degrees of muscle shortening with a rapid stimulus, like a constant contraction or tetanus. And so that's a graded response. It's just there's different degrees of muscle shortening. If I gave you a 5-pound dumbbell and I was like, hey, curl this with your bicep, but stop halfway, you could do that. That's a graded response, okay? You don't have to take it all the way to your shoulder. You can stop halfway and hold it there. That's a graded response. Nothing too terribly tricky. All right, so muscle force, like I told you, depends on the number of fibers that are stimulated. More fibers stimulated, greater muscle tension. No duh, right? Muscles can continue to contract until they run out of energy. And that energy part is what we're going to talk about next time. So we're going to stop here. I'm going to take you back to 3 videos. That's going to be a lot. And I'm just warning you. But take notes, take your time. We'll talk about them more in class, and I'll even pause them to talk to you about them here. So here we go. 3 videos. Number 1, and I can link these on Edmodo as well. There we go. There we go.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open, and calcium...
Okay, so calcium is opening some channels. Quick review. What is the name of the neurotransmitter right in here that this neuron is going to release? Hopefully you said acetylcholine, because that's the one I want you to memorize.
Ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels.
Quick note, we are now on the membrane of the sarcolemma around the muscle. We have left the neuron and we're at the muscle right now.
Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative.
Okay, I'm going to pause it so in your notes you can write sodium in, potassium out. That is important.
Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, so the end was not something you need to memorize, just shows you that it is recycling a little bit here. So next part, to the muscle.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma. and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules.
Okay, so here you have, muscle fiber, muscle fiber, muscle fiber, sarcolemma on the outside. And all they're trying to show you is that in order to get the whole length of the muscle to contract at the same time, the action potential, the sodium in, potassium out kind of thing that I had you write earlier, travels down the sarcolemma, down that plasma membrane, but also in these little honeycomb structures called transverse tubules or T-tubules, and that ensures that everywhere in the muscle fiber is all basically excited at about the same time. That's what they're trying to show you here.
Transverse, or T-tubules, are regularly spaced in foldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm.
Okay, quick note, bear in mind that calcium was the same ion that allowed the end of the neuron to release its neurotransmitter, and now calcium, we're seeing it again, being stored up in the sarcoplasmic reticulum by the billions and billions, and when that calcium is released, it's also what is causing our muscles to contract. So very important ion, calcium.
This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay, last video. And this is arguably the coolest one. Sorry about that.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere.
Okay, so we just zoomed in, way in on one sarcomere. Here's the end of that sarcomere. Here's the other end of that sarcomere. Here's the proteins I told you about earlier, the myosin and the actin. And now we're going to see that calcium that we learned about from the last video come into play in this one.
A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and...
Okay, quick note here. So troponin and tropomyosin are proteins around actin, and at this point in your notes, you haven't seen these guys yet. You've seen the braid. That's the yellow circles here. The braid was actin. You've seen that before. Troponin and tropomyosin are like guarding these binding sites all the time. Myosin is constantly wanting to grab onto actin, but it can't until troponin and tropomyosin move. And so your muscles, think about it this way, they're always wanting to contract, but they have to wait until they get the signal that allows them to, and that signal is the calcium. So calcium binds to troponin, troponin and tropomyosin move, and that myosin head that's been anxiously waiting to grab onto actin can finally do that. And that's what you're seeing in this image here.
Form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate.
Couple quick notes. Myosin cross bridge is actually 2 instead of just 1. Second cool note, it takes ATP not to grab on to actin, but to let go of actin. So if you don't have enough ATP, you can't let go of actin, and that means the muscle can't relax. That's one of the reasons you might see roadkill has rigor mortis, the legs on the dead deer sticking out. The reason that in some crime scene investigation shows or something, they might refer to a dead person as a stiff. It's because when you run out of ATP, rigor mortis sets in, and the muscles all become very hard and very contracted because myosin can't let go of actin. It's kind of an interesting thing.
The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position.
Okay, so it's a lot to take in, but here what we're going to see is a thick myosin filament, thin actin filaments, troponin and tropomyosin being moved, and enough calcium available that they have the signal to contract. And so all we're looking at is how one protein moves another protein, and these guys floating around are hopefully an abundance of ATP.
Binding sites on actin remain exposed. The cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
So once that calcium's gone, we took all the calcium, we reload it back into the sarcoplasmic reticulum for the next contraction, and when it's all gone, troponin and tropomyosin move back. They cover the binding sites, and now myosin, still wanting to grab actin, still having the ATP required to do so, or ADP at any rate, can't do it. There's just nowhere to bind. So that muscle now is able to relax until the next stimulus comes. And so there you go. That's the end of it. So that is where we are going to stop for today. It's a lot to take in, but I'll have a follow along for you to fill out for that. So thank you for your time.